Let's learn about the Calvin cycle. This cycle is also known as the light-independent reactions. That's because it doesn't need light to occur. The Calvin cycle is a series of enzymatic reactions after the light reactions that's going to produce sugar. And this cycle occurs in the stroma of chloroplasts. Don't get too overwhelmed, but this is the Calvin cycle. I'm going to go over the three main parts. The three main parts are carbon fixation, reduction, and regeneration of RUBP. All right, let's start with carbon fixation. When teaching this process, I always tell my students to focus just on the amount of carbon in each molecule. Each one of these purple circles is going to represent a carbon atom. In the first step of carbon fixation, a single carbon dioxide molecule is going to bind with a 5-carbon molecule called RUBP. The enzyme that does this is called Rubisco. Now, Rubisco has a much longer name, but we just call it Rubisco. If you count the carbon molecules in RUBP, there's 5. There's 1 in carbon dioxide, which means this molecule is going to be a 6-carbon compound. This 6-carbon molecule is going to break down into 2 3-carbon molecules. These molecules are called 3-PGA. 3-PGA has a long name, but we just call it 3-PGA. We call this step carbon fixation because we're taking a carbon from carbon dioxide and fixating it into the molecules within the Calvin cycle. The next step within the Calvin cycle is called carbon dioxide reduction. And there are going to be 2 main reactions that take place first. ATP is going to be used up to phosphorylate some of these molecules of 3-PGA, as well as NADPH dropping off electrons into this process. Now, if you remember, ATP and NADPH are going to come from the light-dependent reactions. There is a series of enzymatic reactions that are going to go on here, but we don't focus on those specific enzymes. All that I want you to remember is that these 3-PGA molecules are going to be changed into a molecule that's called G3P. These G3P molecules will also have 3 carbons, and they look identical to 3-PGA, but structurally they are different. The reason why they call this carbon dioxide reduction is that these molecules are going to reduce the molecules here within the process. That's because these molecules, the 3-PGAs, are gaining electrons, which means they're being reduced. In the last main step of the Calvin cycle, we're going to regenerate RUBP. We do this by changing the G3P molecules into RUBP by using ATP. And again, there are other enzymatic reactions that are occurring here, but we don't need to know those. This process remakes RUBP to keep this Calvin cycle going. Now, you might have noticed that one of these G3P molecules is going to go do something different. These G3P molecules are going to be very versatile, which means they can be made into a bunch of different molecules. You can actually take 2 of these molecules and make 1 glucose out of them. But these G3P molecules can be made into a host of other molecules that the plants are going to need. So basically, these G3P molecules are kind of like the building blocks of other molecules like glucose and fatty acids. And it's the main product of the Calvin cycle.